So it's a pleasure to be here. See some familiar faces in the audience. So my name is Haegerstrand, CEO of Annexin Pharmaceuticals since October of last year. I will start talking about the fact that we are actually in the midst of a share issue. We're raising 61 million Swedish kronor at a subscription price of 1.15 Swedish kronor, which will mean, if you multiply it by the current number of shares, not beyond the share issue, but right now the market cap is only about 20 million Swedish kronor. We're in the midst of the subscription period, and you can find the instructions and the prospect on our website, Redeye's website, and also on the Finansinspektionen. The major shareholders are all contributing. 50% of the share issue is already covered by them. Another 50% is covered by guarantors, external, and some other also existing shareholders, and the founders are also involved in contributing to the share issue. So, about the company, established in Stockholm back in 2014, it's a spin-out from Karolinska Institute, where the scientists and the founders still involved in the company found that Annexin A5 is a protein that can not only be used in research to identify dying and damaged cells, it can also be used to treat patients potentially. They started off with a fairly rare disease and proved that in this disease, the vessels in these patients were full of Annexin 5 for a reason, and they found out that this could be potentially a treatment of this disease. So they patented the use of Annexin 5 to treat various diseases. That's really the opportunity we have in the biologics drug field, where you have a use patent, and you can also cover it by production patents. So they started to do that, and we're now a small company based in Stockholm. We're 4 full-time employees, and then we have a number of consultants for specific questions and fields. We are world leading in the field of Annexin A5, and we're the first company to be able to manufacture the drug large scale GMP for even with quality suitable for the market. Focusing on vascular disease, I'll come back to that. And again, our patent portfolio is about the production of Annexin A5 as well as covering the use of Annexin A5 to treat many diseases. There are almost too many opportunities for this drug. It's a really old, we call it ancient protein. It's in all of us here. It's been sitting on the inside of every cell. It's very, very present in the vasculature. So on the inside of the vasculature, including the heart valves, the aorta, the arteries, the veins, the capillaries, everywhere. There's a cell type called endothelial cells covering the inside, and they're really rich in Annexin A5 in the membranes. What Annexin A5 does is when cells get injured, it sort of pops out on the outside and scavenges and binds to certain fatty acids, which are known to be inflammatory and dangerous to the body. So it pops out, binds them, and neutralizes them. Then the whole complex is excreted in the urine. So actually the drug is called, or the protein is called a protective shield, band-aid, missile, bloodhound, etc. So it's really known for that function. What our founders did was to sort of turn this into a therapeutic opportunity. So again, many opportunities for this molecule. We call our molecule ANXV, which is our version of Annexin A5, which is and very, very, very similar to the endogenous natural protein. We're looking at some different kind of diseases. We're looking at smaller indications like retinal vein occlusion, which I will describe in some detail, sickle cell disease. And if we jump to Ebola over here, and even speculations about being possible to use it in coronavirus disease. These are sort of where cells are getting injured in the vasculature. These are blood cells getting clotting, they are sticking to each other, they cause disease. There's a crisis of release of the various inflammatory compounds in Ebola, which can possibly be treated by Annexin A5. Whereas in vascular disease, in the heart and in the vessels, we can really protect also the vessel wall, not the cells inside the vessels, but in the vessel wall. Apart from that, there are also sort of a basic use of Annexin in basic research. Is used actually as a molecular imaging tool. So before we started to work with it, it was used to find damaged cells and damaged areas in the body by tagging a small radioactive compound to Annexin A5, injecting it. And since this radioactive decoy is possible to detect with the camera, you can detect where in the body you have a sort of a diseased cells or dying cells. So we've already been in the clinic, but as a diagnostic tool, or as an imaging tool. You can also tag an anti-cancer drug to Annexin A5 and inject it, and then the Annexin 5 having its missile or bloodhound capabilities, it will find the tumor, find the danger site, and deliver the anti-cancer drug specifically to the tumor, not to the rest of the body. So it's a very smart molecule. Some other companies are considering sepsis, which is blodförgiftning in Swedish, also scarring, other versions of oncology, as well as it's possible to use it obviously also in animal health. So when you work with biologics, you have a challenge as a small company. You have to produce and spend a lot of money and time and effort to produce a drug. Tens of millions of Swedish kronor has been spent on making it your compound that you can put forward into preclinical and clinical trials. So the company has done that for a number of years. We are very proud and happy about the technologies what we have. It's an efficient, cost-effective, high yield, and patented procedure. So we make this in bacteria. We get the source molecule, which we call the drug substance. We dilute it into a solution. It's really an intravenous solution intended in a small vial, which will then get to the clinic. First, of course, obviously you have to have the drug produced, and you have to test it in animal studies, which we have done. This is according to guidelines, but some of you that may have followed the company would know that we had struggles with the findings in the rat. So we have to go think about what really happened. We have to explain why this reaction occurred. We went to the Swedish Medical Agency and asked them how should we deal with this. We got some advice. We went back to the drawing board, made a lot of new studies that were ending in the fall of last year. Now we are ready. We understand why the rats react in the way they do, and we also have another species which doesn't react at all, which is closer to humans. So we're pretty comfortable now that this reaction in the rat is what it is. It's important to follow and understand. But we're comfortable that we can actually follow any such risk in first-in-man studies that are to follow. Clinical development, we will apply for first-in-man trial shortly. We have announced that we do it early 2020. And you start with one single dose, intravenous then, for 1 day. Then you go dose by dose upwards. Then once those single administrations are done, you go with multiple administrations, because we feel that Annexin A5 treatment or Annex V treatment will be for a maximum of 5 days. Intravenous infusion one day, one infusion every day, and that will cover all, in quote unquote, indications that we think are important, because we are wanting to treat this the acute vascular crisis in a patient with an ongoing clotting of cells or with an injury caused to the vascular system. That's where we think the Annexin A5 makes most sense. Clinical selections, and among all these opportunities, retinal vein occlusion, it's a disease that you probably haven't heard about. It's still very problematic to the patients. It's a sudden blindness occurring. So the perspective is almost literally what you see on the left there. The patients go blind, more or less fully blind, all of a sudden, without necessarily any other sort of explanation to it. They get the diagnosis, and the cause of this disease is really a blockage of a vein in the retina. Retina being the area of the eye where the photons are really hitting the body and translated into images. But this area is where all the light is translated into the image eventually. So if you don't have a healthy retina, you cannot see. And if you have the bloodstream with arteries to the capillaries feeding the eye with blood or the retina with blood, and your blockage in the vein that would take the blood back to the heart eventually, the blockage of the vein will cause a sort of a stop in the flow. You will get a back stop, and you have edema and swelling, and then the cells will die in the retina. So you turn blind in the end. So why we focus on this one? I'll come back with some more details. It is the second most common cause of blindness If you don't get the treatment, you have a 10% chance of getting any improvement after you have the diagnosis. With standard of care, which is injecting drugs straight into the eye, you have about 30% to get a meaningful improvement. And there's no current treatment that really addresses the forming of the clot in the vein. Current treatments are really there to stop the swelling and stop the pathological inflammatory process downstream of the initial clot. And we know that the red blood cells from these patients, when they put them in vitro or out in the lab, you can see they clot to each other for some reason which is pretty much unknown. And importantly, I'll come back to the mechanism later, but it's really 28 million people in the world living today that have had RVO at some point in life. Some get a second event as well. Standard of care is very expensive drugs, again, injected into the eye. It can be done one single time every 6 weeks or so, or every month during the course of half a year, that have some improvement of the risk of blindness. It's a huge market. For us, it's definitely enough. It's a $20 billion market. And drivers are, of course, the fact that the people get older and have more atherosclerosis and more diabetes. And we have shown, or others have shown, that if you take these patients' blood cells, as I mentioned, they clot together, but if you put Annexin A5 into the mix, they will not clot anymore in the same fashion. 70% reduction of the actual stickiness. And if you put them together with the vascular cell I spoke about, endothelial, they will not attach to the endothelial either. So we think that by injecting intravenously, Annexin will reach the eye and then potentially even make the clot go away or stop the clot from growing. How we want to move forward with this interesting idea. So the idea would be that we use the current or the expected amount of money that we get from the current share issue to do imaging, phase 1 study, I mentioned that, but also an imaging study in RVO patients. So when patients get into the clinic, get the diagnosis, we will collaborate with specialized teams, with ophthalmologists as well as the imaging specialist. They will have Annexin available. They will attach the radioactive molecule. They will fuse it into the patient. And we can follow then the binding of Annexin A5 in the eye by certain cameras. So we can prove the point that our drug reaches the eye and in the right place in the veins. Next phase, which will be beyond the currently ongoing financing, is to do proof of concept studies. And the eyes, we're blessed with addressing the eye, because in the eye you can actually do a lot of imaging. It's the mirror of the soul. It's also possible to actually look into the eye via the lens. So we can see inside here is the retina and the blood vessels of the retina. To the very right here you can see a blood vessel. You can actually follow the red blood cells in the vessels of the eye. So the hypothesis here is that we will be able to look into the eye. We'll be able to see the blood vessels. We will see the blood cells actually being affected by Annexin A5. And I could not have had a better pre-speaker than the one I had. So we cannot resist the cardiovascular field either. So it's really when surgeons go in and do things in the veins or in the arteries with balloons and what they do. They do good, obviously, but they also injure the vessels. And this injury of the vessels, putting stents into the vessel, mean that they, of course, open the vessel, increase blood flow, reduce the risk of having pain in the leg caused by atherosclerosis or pain in the heart caused by the atherosclerosis. But actually, the stents, there's also quite a lot of problems with them, because the narrowing reoccurs, shown down to the right here, where the stent is there. This is a cross section, and after 6 months, it may look like this. So it's already narrow again. So it is a problem, especially in some patients. This is a very common phenomenon. So given what we know about Annexin, we know that it binds injured cells. So when the surgeons go in to do the catheterization, they will destroy a lot of tissue. This is an illustration of an animal where someone has done, to the left you see nothing almost, to the right you see an injured vessel where we have injected fluorescently labeled Annexin. You see it binds to the injured vessel, and it will then scavenge and protect the cells from the inflammatory process that will come afterwards. Patients with problems in the leg, in the arteries of the leg. To the right here, one patient have been injected with Annexin A5, and you can see it binds to the actual position where the dilatation or the balloon catheterization was done. So, hypothesis here that we will be able to, in the long run, protect the vessels after any kind of vascular surgery being done. Could be heart surgery, could be this kind of vascular surgery. We want to prove the point as a start in this big field. So hypothesis here is that we will know that if you do this kind of procedures, you will have a rise in certain inflammatory markers in plasma. You can take a plasma sample, you will see a pattern like this, going from day 0, 1, 5, 15, it goes up and then it goes down again. And we think that if we have the effect, or that we think with Annexin A5, we will be able to cut this inflammatory effect, which will in turn will reduce the risk of getting back the stenosis or the narrowing. So market overview for RVO, it's a 400,000 patient market, at least for us, which means we're in the $1 million range of sales per annum. We intend to be possibly taking this drug to all through the market ourselves, while for the cardiovascular indications, we think we need to collaborate with larger pharmas. Regarding licensing deals, there was one Annexin deal I'd like to point to, is the one in phase 1 Annexin A1, another Annexin, was licensed by Bristol Myers Squibb for 410 million euros. And also in the ophthalmology field, it's a small field, but still very many deals are done in that field. So we feel comfortable that there are takers out there. A bit of a summary slide, what we intend to do. To the left is where we are right now, what we want to do with the currently ongoing financing or the proceeds of that financing. First in human test or study, imaging in RVO, imaging for another indication, like the cardiovascular indication I mentioned, as well as go to the EMA and FDA and ask them about the development program. You have the luxury of going to them and asking for scientific advice, presenting your case. This is what we want to do, how we want to run the clinical trials. Can you accept that? And the value of having an FDA meeting and the protocol from an FDA, which they have approved the minutes of, it's worth millions, almost literally, for a small company. Not only to be able to describe for investors how to move forward, but also for a licensing partner. They know what the FDA wants. So if beyond that, we want to do proof of concept studies, like smaller concept studies, looking at certain markers, like the blood flow in the eye, like the cytokines in the plasma, and to prove our point before we go to larger phase 2 trials, which are very expensive. Small ones we can do ourselves, potentially. And then, of course, the plan would be to go to final registration for RVO and have multiple licensing partners for the other multiple opportunities for Annexin A5. So in summary, we'll have a first-of-class kind of medicine. There's no other kind of drug that would do the same thing as Annexin A5 for sure. We're the first one to be able to produce it. We have a patent protecting the production. We're in the sort of midst of filing the CTA, the clinical trial application, start the clinical trial this year, be ready by mid next year. We're de-risking the whole development program by imaging and proof of concept studies. And we will also try to grow by potentially partnering some other areas where we're not active ourselves, but we talk to other companies that are using Annexin 5 for other purposes, meaning potential deals. Okay, with that, thank you.
Thank you very much, Anders. So I'll do as usual. I'll turn to the audience first. Any questions? Yeah, one.
I should know this, but I don't. Is there an animal model for RVO?
No.
So how do we then know that this, or not know, but what is the indication that this would work at all?
Sorry. So, lost my voice, drinking too fast. So I guess the answer is there's almost never any perfect animal model for any disease at all. So animal models are always surrogate markers for the real model or the real human pathology. So it's really based on the fact that these patients' red blood cells clot in vitro, they clot in vivo in their eyes, and we can inhibit that clotting with Annexin A5. So when we know the binding between the red blood cells that causes the clot is dependent on this specific fatty acid that annexin binds to and can stop. So that it's a theory in that regard. Another model, which is not RVO, but another one, sickle cell disease, is know that this another type of clotting also dependent on the same fatty acid. In vivo, annexin A5 would so stop that clot from forming. Also. So the hypothesis is based on animal binding data, human blood cell data. That's where it comes from.
You mentioned that you're in an ongoing financing procedure, and you're aiming to raise 61 million Swedish kronor. How far will that get you?
It will take us into mid-2021. So the idea with the whole finding is to reach certain specific milestones, meaning the end of phase 1. So now we can move forward into RVO for phase 2, or pretty much any disease we select, as well as having the imaging studies done as well, in RVO as well as in the cardiovascular disease I mentioned. That will be sort of the basis for potential licensing deals, and also maybe to go back to financiers to raise more money for the proof of concept studies I outlined.
So having an asset that you perceive has such a broad potential in many different indications and within the vascular protection field. RVO is sort of you have narrowed it down and you have a clear idea what you want to do there. What will be next? Is it too early to talk about that?
Yeah, I guess so. I mean, we'd like to work with diseases where the blood cells clot for one or another reason. We can sort of stop that clotting, which goes for RVO and sickle cell disease. Ebola is a bit of a different disease in the sense that you will bind the phosphatidylserine, which is the same fatty acid again, that would cause the storm that causes the dramatic development in Ebola. So we think we can reduce that storm. But then, so I mean, that's one area we would like to pursue for sure. And we start with RVO, probably go beyond that later on. And then is in the vascular field, I think we talked about is large indications enough when we have balloon catheterization. But again, if you think about the fundamentals here, you can protect any vasculature damage to the wall, which surgeons do every day. You know, when they do heart surgery, vascular surgery, I think that's going to be a huge field as well.
Apart from getting the financing done to finance the upcoming studies, then I think you mentioned that there are some small things left to take care of before you can go actually into the clinic.
Well, yes, I mean what we have done is that we've done the animal studies, which we have concluded. We think we have enough evidence. We have to clean, let's call it clean, the drug from a small contamination that we found in the early fall. We're doing that process right now. Of course, then submitting to the Swedish Health Medical Products Agency is not a predictable thing. Really, you can end up getting questions that you have difficulty answering. But given the experience that we have in the company, all the consultants, we feel comfortable that we will get through and to be able to start the studies. So that's really the focus that we have right now to get it through to the clinical study.
Would you say that the slight setback in the fall, finding the reactions in the rats, forced you to take another look sort of at the whole package? Is that now a strength going forward?
Yeah, no, it's a fairly specific thing, right? It was actually discovered almost a year ago. Then we had to go back to the drawing board and design the studies and get them done and get them sort of reported. So it's a fairly specific event, which hasn't led us to rethink anything but that. We have the goal has been RVO. I think we've recently expanded a bit, say not only RVO, but also cardiovascular disease in terms of the imaging studies as a first step.
And summing up, what are the particulars of the ongoing finance and as far as subscription dates and so on?
So it's in the midst of it. Subscription last day is 21st February. I think I mentioned most of it. It's guaranteed in various ways, but I hope all of you will try to subscribe, of course. But it's ongoing. It seems to be going well, although we, of course, as a company, we don't really know.
All right.
The other question.
And one more question? Yeah.
The fact that your product is first in class sounds very nice and interesting, but it's also a major challenge. So what is the major challenge during the next 12 months?
Well, the first in class doesn't really affect you right now. I mean, the challenge is now, of course, that we are sort of moving into the patients with it's not similar to anything but itself. But sort of in that thinking, it has been in man already as a diagnostic tool with the molecular imaging. So at sort of at a very low dose, it has been used in humans already for with the same kind of binding and bioactivity. So it's just the fact that it's different, slightly different molecule with this molecular tag to it. So first in class is normally a good thing. It's a challenging thing when you come to the FDA. You know, they will want to know a little bit more, maybe more about potential drug interactions, etc. And with that, which we'll have to work with going forward in the phase 2 program. But I think there's much value to it as well, because there's nothing similar to it on the market.
Yeah, we're looking forward to see where you take it.
Thank you.
Thank you very much.
Thank you.